Hey y'all, we're going to look more closely at these skeletal muscle models. We have two different ones showing the same things. We're going to look more closely at what components we find within each muscle fiber. Let's start with this one and then we'll move to that one. The outside, the outer layer bordering a muscle fiber, surrounding a muscle fiber, this is the endomysium. This is the endomysium. Just deep to the endomysium is a plasma membrane, and the term for plasma membrane is sarcolemma. So here's the sarcolemma. The sarcolemma has little tunnels that allow the membrane to go deeper into the cell, into the fiber. And these tunnels wrap around myofibrils. You can see in blue, these tunnels wrap around individual myofibrils. These are T-tubules. These tunnels are called T-tubules. T is for transverse. They're going transverse through the muscle. T-tubules help spread the charge more evenly deeper into the muscle, so you get activation of all these myofibrils, not just some of them. Here in this yellowish color, that's the sarcoplasmic reticulum. That is what releases calcium to the sarcomeres to control contraction. So if you remove all that and look at the individual sarcomere, what we can find are the sarcomeres. So this, from here to here, that is one sarcomere, here's another sarcomere, and so on. It's actually represented better, in my opinion, on this cardiac muscle model. It shows you more clearly, here's the M line, here's the Z line on either side, and this is the sarcomere. And it's bordered by T-tubules, surrounded by sarcoplasmic reticulum. I prefer showing sarcomeres on this model or just showing the whole sarcomere model. Once again, M line, the dark green ones are thick filaments, so the lighter ones are thin filaments. Z-line, Z-line. Lots of mitochondria, because resetting muscle, getting it to contract, requires lots of ATP for reasons you'll discuss in physiology. And once again, multiple nuclei. I only found one right now. There should be more, but these are really long fibers. It's a multinucleate cell. On this one, here you can see the endomysium, sarcolemma, nucleus. I don't see mitochondria as much on this one. Myofibrils, sarcomeres, you got your M line, Z line, M line, Z line, M line, Z line. The darker is thick filament, the lighter is thin filament. Or I guess the A bands and the I bands. A band, I band, A band, I band. Back to this model, here we have a cut-off axon of a motor neuron. This is a myelin sheath. We'll get into what that is later, but this is the layers of myelin surrounding the axon. So really the yellow here is the axon of the neuron. The axon of a neuron is what sends a signal, and then it will eventually control the muscle fiber. So here's our axon terminal in yellow. In the axon terminal, these white dots are representing vesicles. Those are vesicles full of neurotransmitter. When the axon sends a signal, the vesicles are released by the neurotransmitter within vesicles are released by exocytosis. And this gap here, it's called a synaptic cleft. This special synapse, a communication area between a neuron and something else, this special synapse between a neuron and a muscle is called a neuromuscular junction. This is a neuromuscular junction. So neurotransmitter gets released into the synaptic cleft. It'll bind to special receptors on this squiggly part of the sarcolemma. This is the motor end plate. It's called something different because this is where we have receptors for this neurotransmitter. So neurotransmitter binds to receptor on motor end plate. It opens up a channel, a transport protein, and lets sodium into the cell. So now we have sodium flooding the sarcoplasm. And it can spread deeper into the cell, surround the myofibrils with the help of these T-tubules. It's spreading along the T-tubules, going deeper into the cell. So we're getting all this charge getting into the cell, deeper in. What that charge does, it activates the sarcoplasmic reticulum. The sarcoplasmic reticulum, when activated, releases calcium into the sarcoplasm. When calcium is in the sarcoplasm, calcium binds to troponin. Troponin moves tropomyosin out of the way. It allows myosin to bind to actin and allow for the power stroke to happen. We get contraction. Once again, myelinated axon, axon terminals, neurotransmitter released into the synaptic cleft of the neuromuscular junction, binding to the squiggly thing, that's a motor end plate. Axon terminal, there should be an axon terminal here, but it's been cut off, so you just see the end of the axon. This is the motor end plate where you find receptors. Receptors get activated, open up a transport protein, a channel to allow sodium to flood into the cell. Sodium floods in, travels along T-tubules, activates sarcoplasmic reticula, which isn't shown in this model. Sarcoplasmic reticula release calcium, calcium binds to troponin, troponin moves tropomyosin out of the way. That allows myosin to bind to actin, complete the power stroke. You'll have several contraction cycles for contraction. Those are our skeletal muscle models. This one shows a bit more detail. This one shows the banding a lot more clearly. You can see the A bands. A bands are the darker ones. I bands are the lighter band. I band, A band, I band, A band.